Okay, the next speaker of this session is Yigu Zhou from Montreal Neurological Institute, Canada. The title of the presentation, the local tissue and hemodynamic properties reflect pathological discharges in the epileptic brain.
Hello, everyone. Yeah, so we're going to talk about epilepsy today. So epilepsy is one of the most common neurological disorders worldwide. And the main concern of neuroscience research in epilepsy is this concept of epileptogenesis. We can break it down in its Latin roots of the word, but basically it's the idea that there has to be something wrong with structure, function in the brain, in some brain area, in order for seizures to occur. And seizures are these very large-scale events that are like embodied. So the gold standard for the epileptogenic zone in the brain is so far still like relying on post-surgical outcome, which means to really know if a part of the brain is epileptogenic, you have to remove it and then see if the patient still has seizures. If not, then the part of the brain that you've removed definitely contains the epileptogenic zone. That's the hypothesis. But in order to have someone agree to undergo surgery, you have to have a best guess before you take a part of their brain out. And electrophysiological studies and imaging complement each other in the research and diagnostic workup for these surgical patients. So we've established that there's like a interaction of different components for normal brain function that is non-seizure inducing. So we can measure different aspects of tissue and hemodynamics with neuroimaging and more finer scale of electrophysiology with something that is highly temporal sensitive and spatially specific, like intracranial EEG. But how do these different compartments of brain structure and function interact with each other in generating seizure-related pathology is a question that I want to answer, and I'm very lucky to have my hands on this incredible surgical candidates data set. So these are patients that are admitted at the MNI, and they come in with recurrent seizures, suffering greatly from it, and they were scanned before they were implanted with deep electrodes. So the scanning protocol is like a 3T protocol. This protocol has been published with healthy controls before in the Mika mix data set, and it was pre-processed with Mika pipe. The measures that we're interested in are quantitative T1 measures that are myelin sensitive, diffusion measures, and hemodynamic assessed with resting state BOLD. So I'm going to tell you about these tissue measures first. We have 5 measures associated with QT1 are the first 3 statistical moments of the depth distribution of myelin intensity, basically assessed with T1 relaxometry. So we can generate a histogram across depth at every single vertex sampled with these equidistant surfaces. And the mean, so M1 is supposed to be cellularity, so the mean value of QT1 intensity across the depth. M2, variance, it reflects the columnar differentiation of myelin distribution, and M2 is supposed to be how much, like the myelin distribution skews toward a certain layer, and that tells us about columnar organization. For diffusion, we have a fractional anisotropy and a proxy for mean diffusivity, which is ADC. And for BOLD, we took these time series and extracted 22 time series features using the multidisciplinary library of Catch21, which is like a abbreviated version of HCCSA that is minimally redundant in the features that they capture. And for electrophysiology, each patient was implanted in different places of their brain based on their symptom. And when I concatenate them together, they kind of offer a pretty good whole brain coverage on the right hemisphere, and then I just kind of like mirrored them so that they live on the same hemisphere. Historically, so there is a long history of using electrophysiology to locate potentially epileptogenic zones in the cortex. We cover several interictal and ictal measures. So these interictal measures are taken when the patients are not having seizures during non-REM sleep. And ripple events and fast ripple events are these high-frequency oscillation bursts. We all have them. They subserve physiological functions, but when they exceed a certain either region-wise or whole brain thresholds, they are considered pathological. And for ictal measures, so I had to rely on some medical expertise here that really looked at the seizure recordings and labeled channels that are like in regions of the brain initiating the seizures. So these features overlap with each other in different extent, meaning they don't all index the same electrophysiological abnormalities. And when we compare electrophysiological markers with imaging markers, we can see non-overlapping sets of hemodynamic changes. These are just univariate comparisons between pathological and non-pathological channels, with the exception of seizure onset zone being more differentiated by M2, which is columnar organization, by QT1. And here I'm assessing whether tissue and hemodynamic changes can be used as biomarkers to differentiate pathological and non-pathological channels by electrophysiological standards. And we can see tissue overwhelmingly predicts, overwhelmingly overperforms the other sets of features in distinguishing pathological and non-pathological channels, with the combined model kind of doing a little better than the tissue-only model. So now I ask, how do they interact with each other, tissue, hemodynamic, and electrophysiology? I'm going to focus on ripple rates for now. This is a mediation analysis because here we know that there's a multivariate interaction between all of these individual features. Now I want to know what are kind of the directionalities of these interactions. So this is the setup of the mediation model. We can discuss this later if you want to ask the specifics. But basically what I'm really interested in is the mediation effect that indexes how much the mediator participates both in predicting the X, which is the input, and Y, the outcome. And I've iterated all combinations of paths, so not assuming anything, because we have a wide range of tissue and hemodynamic measures. I just did a PCH so that I have a single factor for each. I did a weighted sum of all the significant components and yielding these over 70% various explained factors in my mediation models. So these are all the combinations that I've tested. We're going to go through them like pairs of them at a time. So between tissue and hemodynamics, if I use the direction of the arrow goes from X to Y, and the square box in between is the mediator, we can see that tissue and hemodynamic features totally and directly predict each other without a mediation of electrophysiology. And here, hemodynamics and electrophysiology also totally and directly predict each other without the mediation of structural factors. And finally, structural changes require the mediation of hemodynamic changes to predict ripple rates. So in summary, I took advantage of this clinical data set to integrate multiple modalities to see the potential biomarkers and mechanisms of pathophysiology in these epilepsy patients. And we found that with the logistic regression, tissue changes most accurately distinguish healthy versus pathological cortex, although tissue changes did not very much relate to fast ripples and mostly just ripples. And with the mediation analysis, we found this feedback loop that really put hemodynamic factors at the mediator role between the relationship, in the relationship between tissue and ripple rates, which directly and totally predict both tissue changes and ripple rates. So I'm going to thank my supervisor and the clinical expertise, people who refer patients to us also, and lab mates. Thank you very much.
Thank you very much. Are there any questions to the speakers? There's one just question coming to the up. So how did you adjust for other input for NTS such as pulmonary stretch receptor hypothalamus in your study?
Sorry, hypothalamus?
So I don't know this is what it's coming from, but maybe, I don't know. The person is just asking maybe some subcortical or hypothalamic just influence to some epilepsy heart rate. Is that just considered in your studies?
Yeah. So working with intracranial EEG data has been like pretty challenging in terms of registration. But as far as so the location of the intracranial EEG channel isn't really a determinant for the outcome of the relationship between these various measures per se, because they're all sampled in the same spatial vicinity. So we didn't look at hypothalamus or subcortical structure per se, but also most of these patients have like cortical. Well, surgery can only be conducted on cortical regions, like when you resect, so that's why.
So I have a quick question. So you have just many variables for tissue, like maybe mean diffusivity, FA or QT1, then you're just doing multivariate analysis. I'm just kind of wondering, then do you have any just idea about which just variable contribute more than the others? So are there any observations?
We do. I can show you the PCA components if you want.
But just quickly. So.
We have an overwhelming representation of different myelin-sensitive measures. So I have it here for the regression, logistic regression, but I didn't include it for the PCAs. But basically, both variance and skewness contribute, like for the interictal measures. But it's really based on, it's really reliant on their interaction with the rest of the hemodynamic changes.
Thank you very much for the interest of time. Let's move to the next speaker. Thank you very much.
Thank you.